Hello, everyone, and welcome to today's panel presentation, a blood test for Alzheimer's disease, thinking beyond a diagnosis. This webinar is part of the 12th annual event in the Neuroscience Virtual Event Series. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Alamar Biosciences. For more information about Alamar Biosciences, go to alamarbio.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. I now present today's speakers, Nicholas Ashton, Associate Professor of Neurochemistry at the University of Gothenburg, and Alex Forrest-Hay, VP of Sales at Alamar Biosciences. For a complete biography on our speakers, please visit the Presenters tab. Nicholas and Alex, you now may begin your presentation.
Thank you very much for the introduction. Really excited to be here today to record this webinar for LabRoots and introduce you to Alamar Biosciences and the NULISA technology. NULISA is a nucleic acid-linked immunosandwich assay. We recently launched the platform, and it's a fully automated ultra-sensitive proteomics platform for profiling biofluid samples. Alamar Biosciences was founded in 2018. The mission for the company is really to enable ultra-sensitive precision proteomics to enable the earliest detection of disease. And there's been three major innovations that have been pioneered by the Alamar team. The first is NULISA, which I'll talk a lot about today, which is an ultra-sensitive atomolar level NGS-enabled digital immunoassay platform for protein analysis from single proteins up to potentially thousands of plex. We've also developed a novel antibody technology called AtoBody, and these are biparatopic antibodies with picomolar affinities, which we can leverage for developing ultrasensitive assays for high priority biomarkers. And thirdly, we've developed a fully automated instrument called the Argo HT. And this is a fully automated system, benchtop system, that performs the NULISA assay across a range of different multiplex levels and enables you to do this with minimal hands-on time. So you can truly go from discovery right through to clinical implementation. So really the mission for Alamar is to provide automated solutions for ultra-sensitive protein analysis across a range of different multiplex levels. The company is based in San Francisco Bay Area in Fremont, and we've fully commercially launched the platform and recently secured additional funding through a Series C raise of $128 million. So really, we believe at Alamar that the power of the NULISA platform comes from the greater sensitivity that we can provide. And this sensitivity enables you to then measure important biomarkers at lower limits of detection and quantify them accurately. And this is very important when you're looking for early markers of disease. But equally important is you can actually now discover novel biomarkers that are present at very low levels in patient blood samples to enable early understanding of disease and earlier detection of diseases. And what we strive for with NULISA is to be able to detect protein biomarkers in healthy individuals as well as pre-symptomatic or early stage disease individuals. And this sensitivity enables us to really understand and get a clear understanding of a healthy baseline from human plasma samples. So at the highest level, there are really four pillars that we live by at Alamar with the NULISA assay. The first of all is unprecedented sensitivity. So being able to measure multiple biomarkers at the same time at atomolar levels of sensitivity is really unprecedented. And this enables you to quantify very low abundant proteins and enables the discovery of critical biomarkers. We also have a technology that has the widest dynamic range. So we can actually measure proteins across 12 logs of dynamic range which is one of the really biggest challenges of plasma proteomics, is the range of proteins that are present within a normal plasma sample. And this means we can quantify proteins across a whole biological range without the need for any sample dilutions. And fundamentally, NULISA is a very flexible chemistry, so you can measure single biomarkers, or you can develop large panels and measure them with the same sensitivity. So we have off-the-shelf offerings for very critical biomarkers like p-tau217, at the same time panels of biomarkers to measure biological pathways in different diseases. And finally, we've fully automated the platform, so it's taken all of the arduous manual steps away from generating data, and is a very easy to use platform with very high reproducibility. So just to briefly introduce you to the Argo HT instrument, you can see the instrument here. It's a benchtop instrument which has 3 bays to enable you to run 3 96-well plates at a time. And Alamar provides consumable kits that provide all of the reagents and plasticware required for a run, whether it be a panel or a single biomarker. And we also provide on the back end software to enable both sample setup, QC of the data, and then secondary analysis. So this is really proteomics now at the push of a button. You don't need any additional equipment to run the assays. You literally load the samples and walk away. So the HT system, actually, to set up a run on the system, takes less than 30 minutes of hands-on time. You load the samples in a 96-well plate, and then you load the cartridge of consumables, and then you walk away. Singleplex assays on the data for the singleplex assays are generated on the onboard qPCR instrument, and the data comes out of the instrument after 8 hours for each plate. So the turnaround time for a single plate is 8 hours. And if you're doing multiplexed assays, so hundreds of biomarkers simultaneously, then the readout is done using a Illumina NGS system. And the Argo instrument takes you right through from sample through to an NGS-ready library that you then load onto an Illumina sequencer for the generation of the data. So this is really a high throughput instrument where you can run 3 96-well plates a day with no manual steps. Once you've started a run, it's walk-away automation. And the sample input for plasma and serum and CSF samples is between 10 and 20 microliters. And the integrated data analysis is done using a cloud-based analytics software platform. So really the Argo HT is enabling both scalability and flexibility. So we have panels to enable discovery, where you can look at hundreds of biomarkers with very high sensitivity for focused discovery within sort of known biological systems. Then you can translate these discoveries with targeted panels or individual biomarkers to translate these interesting findings into potential clinical assays, and then ultimately run them in the singleplex or lowplex mode using the qPCR system that's on board within the Argo for future diagnostic applications. So just to dive into how the NULISA technology works briefly and how we achieve such high specificity and sensitivity using this technology. So we used monoclonal antibodies that are sourced from commercial partnerships that we have, which are designed to target different epitopes on the proteins of interest that you're trying to measure. And then to these 2 monoclonal antibodies, we conjugate oligo sequences with a known specific barcode for that antibody. So each antibody has its own barcode that can be read out either by qPCR or by sequencing. What's really unique about NULISA is that we have these double-stranded sequences on each of the antibodies, one that ends with a poly-A-tailed oligo and one that ends with a biotin. And those are what enable us to actually purify the immune complex and remove background to improve the signal to noise and achieve exceptionally high levels of sensitivity. So the combination here of a dual hybridization event and the sequencing readout, and then the capture and wash steps, makes NULISA the most sensitive and specific platform currently available. And the steps of the assay are really very simple. It's all automated on the Argo, but essentially the first step is the incubation of the antibodies as they bind to the proteins of interest. Then oligo DT beads are added to the sample, and the poly-A tail on the first antibody bind to the oligo DT beads, which enables us to do an initial wash step to remove matrix, unbound proteins and unbound antibodies. We then release that back into solution and then add streptavidin beads, which enables us to capture the immune complex for a second time using the second antibody and the biotin. And this enables us then to wash away any remaining antibodies or unbound antibodies and do a second purification step. So we have a very clean immune complex. And then at this point, we do the ligation, amplification, and then the measurement. So by doing these sequential capture and wash steps, it really enables us to remove background and improve sensitivity significantly. So the advantage of this sensitivity and this sequential capture and wash step is shown here. On the left-hand side, we're comparing the IL-4 assay generated on a standard PLA assay with the NULISA technology. And you can see with the blue line that we are able to achieve about 10,000-fold higher sensitivity with NULISA than you do with a conventional PLA assay. And this is because of the removal of that background and the improvement of the signal to noise. But what's also important here is you can see that we can achieve 8 logs of dynamic range for this assay, where you can quantify IL-4 at very low levels and also very much higher levels across 8 orders of magnitude. So this is a really significant enhancement over conventional PLA methods. And on the right-hand side, we're comparing it with the current best-in-class sensitivity platform from Quanterix, Simoa. And here we've developed an assay with the same antibodies with NULISA for HIV P24 protein and compared it directly to Simoa. And you can see that here we're able to achieve about a 10-fold improvement in sensitivity over the current highest sensitivity platform and a broader dynamic range. So we really started off by focusing on very low abundant cytokines, which are very important markers, obviously, of inflammation and immune response, and typically very hard to measure in human plasma samples, particularly healthy individuals. And here you can see the really high sensitivity that we're able to achieve for 6 important markers that are typically very hard to detect with conventional technologies. And we're down in the femtogram per mil, single-digit femtogram per mil levels in terms of limit of detection with the NULISA technology. And when we do develop assays, for example, this IL-1 beta assay, you can see that we go into a lot of rigor to validate the assays, not only looking at LOD, but we also look at analytical lower limits of quantification, and we always strive to get 100% detectability in healthy plasma samples with all of the assays that we develop. And here you can also see that we've done parallelism analysis and looking across 16x dilutions to make sure that we're getting a linear response, which shows that we have a very precise and robust assay. And we also do spike recovery to make sure that we're not getting any matrix effects and that we have a very high performing assay. So overall, we have very high sensitivity, very high accuracy, and very robust technology for detecting low abundant proteins. And recently, we've really turned our attention to neurodegenerative markers, because there's a huge unmet need, obviously, for detecting low abundant markers in plasma samples to look for neurodegenerative diseases. NFL is one of the flagship biomarkers. And here we're comparing our NFL assay to the current Simoa assay. And what you can see here is we have a high sensitivity, we have very good correlation with the current gold standard, and we have a broader dynamic range and utilizing 5 times less sample. So we've really gone and developed an NFL assay, which is best in class. And we've also developed phosphorylated tau 181 assays with exceptionally high sensitivity and very good performance across a range of sample types, serum, plasma, and CSF. And probably the hottest marker right now in Alzheimer's disease is phospho-tau 217. And here we're showcasing the sensitivity that we can achieve with our NULISA assay, utilizing antibodies that we source from a partnership with AlzPath. And here we have single-digit 6 femtogram per mil limits of detection, very low functional LODs, and very, very good metrics across the different metrics that we track for performance of the assay. So we're very excited about the p-tau 217 assay being best in class from a sensitivity and performance standpoint. So we have a number of singleplex assays that are available and can be run directly on the Argo instrument, and you can see them listed here. We've focused mainly on neurodegenerative and inflammatory markers that are very typically very difficult to measure, but important from a biological context. The important thing about these singleplex assays, they have a very broad dynamic range, very high reproducibility, and very high sensitivity. Perhaps the most exciting thing about NULISA is that we can achieve these high levels of sensitivity not only by looking at individual biomarkers, but by looking at panels of biomarkers. And here we're just showing 5 cytokines that have come from LPS-stimulated whole blood samples, and we've measured them with the singleplex qPCR assay as well as a multiplexed assay using the NGS readout. And the results are effectively the same. This means that we can build panels, and the first panel that we've built is this inflammation panel, 250 biomarkers that can all be measured in 10 microliters of plasma or serum or CSF with very high sensitivity across a very broad dynamic range. So it's now possible to look across a very broad range of inflammatory and immune markers, cytokines and chemokines with a very small amount of sample with very high sensitivity. And there are many novel biomarkers in this panel that have previously been undetectable with other technologies, like all of the different IL-17 markers, for example, interferons, different growth factors, and TNF family markers that are unique to the NULISA platform. So we believe this inflammation panel is really best in class for studying inflammation from human plasma samples. And we recently published a paper in Nature Communications. Please take a look at this, as it really showcases the data and showcases the power of the platform, but it also compares it to current platforms that are available, such as Olink, MSD, and Quanterix. And in this publication, it shows that we're able to achieve very, very high detectability for all of the markers in the panel, which is actually very unique to be able to actually measure all of these 250 biomarkers in healthy individuals with very high detectability. And when you look at detectability across different platforms, you can see that NULISA has the best detectability compared to, in this case, the Olink Explore and MSD. And the correlation is generally very high between the platforms, apart from in those within those biomarkers where detectability is low in the MSD and the Olink platforms. So we're showing very good correlation and very good performance. And on the right-hand side, we're also showing very, very good CVs, both intraplate and interplate CVs, typically less than 10%. And why is that sensitivity important? Well, when you look at it from a biological context, looking at inflammation markers, usually you can detect them quite well in patients that have inflammatory response, but it's very hard to detect them in healthy individuals. And in this study, we actually compared directly the Olink Explore panel with the NULISA inflammation panel. And what you can see is that there were a number of biomarkers here in the middle that were statistically significantly changing in patients with inflammatory disease versus healthy donors. But there are also a very large subset of important cytokines like IL-4, IL-32, IL-6 that were differentially expressed, but were not detectable or not seen to be differentially expressed with the less sensitive platform from Olink. So really what you're able to see with high sensitivity is the true underlying biology that's really driving disease because you can measure these samples both in healthy individuals and in disease samples. So we've also validated this technology in dried blood spots, which is a very hot area and very important. So we can do at-home collection of blood samples and measure low abundant proteins with very high accuracy. And this just shows that 82% of the proteins on the 250-plex inflammation panel are detectable in the dried blood spot samples, which is the highest of any platform that's been published. And these include markers like IL-33 and IL-4, which benefit from the very low limits of detection, and so you can actually measure these in dry blood spots for the first time. So moving into neurodegeneration, which is really a really big focus for this meeting, of course. Here we have a new panel which we just launched called the NULISA-Seq CNS Disease Panel 120. And this was designed with input from pharmaceutical companies and academic KOLs like Dr. Ashton, who really were looking for a panel that covered as many of the neurodegenerative biomarkers and the neuroinflammatory biomarkers as possible. This has previously not been possible to develop a panel like this because you need very high sensitivity and high plex to achieve the measurements. So you can see here that you can actually measure all of the different p-taus, NFL, TDP43, amyloid beta 42 and 40, as well as all of these important cytokines and chemokines within a single experiment with 10 microliters of blood. So this is really enabling you to look very deeply and very broadly at all of the key neuroinflammatory biomarkers that drive CNS disease. We can do this with very high reproducibility with typical CVs, less than 10%. And in plasma samples, we're able to detect the vast majority of these markers in 100% of plasma samples that we measure. In this particular case, we're showing that 116 targets out of the 120 114 targets out of the 116 were detectable in these particular samples. And in CSF, we also see about 88% detectability of all the markers in the panel. The majority of the neurodegenerative markers are detectable in 100% of CSF samples. So being able to do this really enables you to measure important biomarkers like p-tau217 in both plasma and CSF. And this data here really shows p-tau217 can be measured very accurately in Alzheimer's disease patients and non-Alzheimer's disease patients. And you can measure them either on the y-axis, we're showing picogram per mil levels measured with the qPCR assay, the singleplex assay, and on the x-axis using the multiplex panel. And you can see an R-squared of 0.97, showing that essentially you get the same data and the same sensitivity, whether you measure this as part of the panel or whether you measure it as a singleplex assay. So this is quite remarkable that you can now measure 120 biomarkers with exceptional sensitivity, with equivalent sensitivity to singleplex markers. And when we do this in patients with Alzheimer's disease and compare them to healthy controls, you can really start to unlock the biological understanding of Alzheimer's disease. And you can see the key biomarkers that pop up here, like p-tau217, p-tau231, amyloid beta, key biomarkers of Alzheimer's disease. And we've also run subsets of samples for Parkinson's disease. And you can see that we can measure things like alpha-synuclein and TDP-43 and really get a fuller understanding of what particular biomarkers, blood-based biomarkers, are driving neurodegenerative disease. So I'll leave it there. Just to summarize, we have a very high sensitivity platform with a very broad dynamic range and a fully automated instrument to process either single biomarkers or panels of biomarkers. And we have the Argo instrument is designed to be super easy to use and enable you to run high throughput projects with minimal hands-on time. We also have a technology access program where you can send samples to Alamar and we can generate data. And that's something that we've done with the University of Gothenburg. And so what I'm going to do now is hand over to Dr. Nicholas Ashton, the Associate Professor from the University of Gothenburg, who can present some of his data that he has generated using the NULISA platform. Thank you, Dr. Ashton.
Thank you, Alex. It's great to be with you today. And what I would like to present in this 30 minutes or so is kind of the landscape for a blood test for Alzheimer's disease and the amazing progress that we've made in the last 5 years and how a technology like Alamar is going to advance this a little bit further. So Alzheimer's disease right now is essentially turning towards a biological disease. Not only do you look at the clinical symptoms of the patient, but also now we're looking at the underlying pathology. And the first really biological definition was by Alois Alzheimer, where he described his patient, Augusta Dieter, in 1906 with these extracellular plaques and intracellular tangles. And it was till the mid-80s and early 90s that we knew that these contained proteins of amyloid beta and tau. And moving to today, or at least in 2018, we actually introduced a biological criteria into research for Alzheimer's disease, and this is being more adopted into the clinical space, where we can actually identify these amyloid plaques and tau tangles by imaging and cerebral spinal fluid biomarkers, and neurodegeneration, so the death of the neuron with different imaging, again, and CSF biomarkers. And this became our ATN criteria that we use in Alzheimer's. And Alzheimer's disease really focuses on amyloid, one of these proteins. It's the protein that deposits first in Alzheimer's disease patients. And we have very exciting advances in drug development. We've had one drug or two drugs approved in the United States, or FDA approved, and another one on the way, which clears amyloid completely from the brain of these patients. These are high-level results from these individuals. You can see from the decreases in the plots on both of these drugs called donanemab and lecanemab, completely clearing amyloid from the brain of these individuals after a year and a half or 2 years. Now, these drug trials were targeting people with mild symptoms, but Alzheimer's disease is actually a continuum of pathology build-up, which can actually start before you have symptoms, and we call preclinical Alzheimer's disease. So what I want to show you here in this next slide is an amalgamation of familial Alzheimer's disease cases. So these are rare type of genetic Alzheimer's disease, and show you the amyloid buildup that happens in these individuals over the preclinical space. So we'll start off here about 20 years before the symptom onset, and I just want to show you the amyloid buildup in this brain. Okay, I've paused it here essentially at 0. So we can see that over these 20 years, there's a whole lot of amyloid that's been developing. It's actually saturated in certain areas of the brain. And it shows us that a lot of amyloid is developing before even symptoms occur. Now I'm going to continue the slide, which is when someone has been given their diagnosis, they're starting to show symptoms. And I want to look at the amyloid deposition from this point. Okay, so hopefully what you've been able to appreciate from here is that after symptom onset, there was essentially a very little amyloid change in this brain, really illustrating that Alzheimer's disease pathology in terms of amyloid starts before the symptoms occur. And this is where biomarkers become extremely important, and particularly a blood biomarker, because it's easily accessible, becomes very, very important to try and catch individuals much earlier. So a blood biomarker, really, I see it as 3 key distinctions: diagnostics to help a clinician come to a quicker, more accurate decision about the patient that's in front of them. We need to be able to monitor patients that are on investigative drugs or approved drugs. And for us as researchers, having a tool for us to explore new hypotheses, but able to use a blood test to characterize volunteers or research participants is extremely valuable to us. But the idea of a blood test is not a new one. It's actually something that I've been working on over a decade, and people before me there. And this is actually 2 headlines from a newspaper in the UK. The same newspaper, very similar headlines, but one was in 2014 and the other was a few weeks ago in 2024. So what's the difference between these? There's a decade between these studies, and why are we so excited about this now and not a decade ago? So I just want to look at brief, quick run through of proteomics on a blood test. So this is one of the first studies in 2006 that looked at blood in Alzheimer's disease patients. And we use this technology called 2D gel electrophoresis, where you separate proteins on a gel based on their pH and their molecular weight. You looked at the intensity of the spots on this gel between an Alzheimer's disease patient and someone who was healthy. The spots which were more intense were cut out, they were digested, the proteins extracted and run a mass spectrometry and identified those proteins. And then you inferred that these were the proteins that were changing were significant. So it was a long process, a very stressful process for me when I was doing my PhD. But even then in this process, we could reject the null hypothesis. We could detect differences in the blood between Alzheimer's disease patients and controls, and these pointed towards inflammation, the complement system, and the complement cascade. Coming into 2014 and further on, we started to have new mass spectrometers, new ways to multiplex blood and separate proteins, and we were able to build these panels of proteins which could identify Alzheimer's disease patients, or in this paper that I'm showing on the right-hand side, amyloid pathology versus controls. And there were different ways that we used proteomics to do this. And usually this was a non-targeted route coming down the right-hand side to find a specific target, and we would use more specific targets when we have more specific analysis when we had a target in mind. And here on this slide, what I want to illustrate is all the suggested blood biomarkers that have been proposed in Alzheimer's disease, kind of congregated on this top of the slide here. And they all correlate with each other, but they haven't been replicated very widely. And as you see from down on the bottom is now what we call our core biomarkers for Alzheimer's disease in blood. And there's very little correlation between them, shown by the intensities of these connection lines. And really the biomarkers that we've been looking at in our proteomics for the last decade reflect the periphery, and probably something secondary to having an Alzheimer's disease diagnosis by having poorer health, kidney disease and liver disease and other things. And really we want to have the specificity to identify biomarkers that reflect the pathology. And this is where we are today, and I've called this kind of section the biomarker revolution. And this on the left-hand side is what we can do in cerebral spinal fluid in clinical chemistry routine here in Sweden and many people around the world. And then blood is the biomarkers that we can detect using technologies today. And we're catching up and essentially can do very similar things, not everything, but very similar things in blood. And I will be talking about phosphorylated tau, but I want to mention 3 other biomarkers that we are looking at in terms of Alzheimer's disease. First one is neurofilament light, which Alex had mentioned before. And this is a biomarker of general axonal injury. It increases in all neurodegenerative disorders, as you can see on the left-hand side. It's more increased in ALS and Down syndrome dementia and frontal temporal dementia. And so it's not a specific biomarker, but it's a good indicator of injury. In the middle here, we have cutoffs based on age. So this is a biomarker that increases with age, even if there's no disease present. And on the right-hand side, actually, neurofilament light is a very good indicator of prognostication in cardiac arrest. So if you are unconscious because of an out-of-hospital cardiac arrest, high levels of neurofilament light indicate severe brain damage, and after 6 months, you're less likely to have made a full recovery from this brain damage. Whereas low levels, even though you are unconscious and to a clinician may seem very similar to a severe case, you're more likely to make a good recovery. So it's a great way to triage in that way. GFAP is another biomarker. It's an astrocytic biomarker, which we can measure in blood. And this is very, very related to the development of amyloid pathology. So as amyloid develops in the brain, even if there's no symptoms, GFAP starts to increase. And we know this from familial Alzheimer's disease, and also know this from Down syndrome patients as well. Interestingly, and this is a well-replicated finding, that GFAP in blood is a better biomarker of the brain than GFAP in CSF. We don't quite understand the reasoning behind that at the moment, but that is a very well-replicated finding. And on the right-hand side, of course, GFAP is a biomarker of astrocytes. How much does it reflect astrocytes in the blood is an open question right now, but we're starting to see that actually it does reflect astrocytes, but astrocytes that are reacting to amyloid pathology rather than any other proteinopathy. And lastly, amyloid beta, and this has been a difficult biomarker to measure in blood because there is a lot of peripheral amyloid expression. CSF Aβ42/40 is our core biomarker for Alzheimer's disease. This is what our diagnosis is pinned towards. And we do have good assays that work very well in blood, but Aβ has an issue in blood in that it has a very, very small fold change between Alzheimer's disease and non-Alzheimer's patients. It's only around about 10%, where it's about 50% in CSF. So if you look at these bimodal curves in the center here, you can see on the left-hand side, this is CSF. Very easy to draw a cutoff, two very clear distinct groups. On the right-hand side, this is plasma. There are 2 distinct groups there, but you can see that the overlaps is large and the difference between the 2 groups is very small. So this is going to be a difficult biomarker to use in clinical routine. And lastly, peripheral Aβ is affected by pharmacodynamic effects like Entresto. So this is a heart failure drug. It's used by 1.5 million individuals in the United States, and Entresto is a neprilysin inhibitor. So this acts on Aβ. So what you will see is individuals with these drugs, they will have lower Aβ42/40 levels because of this drug and not because of Alzheimer's disease. But if you didn't know that, these would be classified or have a molecular diagnosis of Alzheimer's. Phosphorylated tau, NFL, GFAP are unaffected by these pharmacodynamic changes. So I briefly want to talk about phosphorylated tau, and it's impossible to summarize this data even in a 5-hour talk because there's so much. But I just want to put it in 5 generations for us to see what has been done in literally 5 years in this field. So in about 2018 and 2020, and there's a few smaller papers before this, we started to develop these ultra-sensitive methods, mainly on the Samoa, but also mesoscale as well, showing increases of phosphorylated tau in the blood. And these were validated against amyloid PET and also are gold standard in neuropathological findings. I would call these proof of principle, they're definitely pioneering, but you can see here that the box plots here still have large overlaps. I would still class these as academic assays, far away from being clinical use, but they were a major step forward in our development. And in 2021, we started to see a slight change. So we started to discover that multiple epitopes of phosphorylated tau existed in blood, like 231, and more importantly, phospho-tau217, which is developed to be our principal biomarker in blood. In the center, we show a landmark paper by the Biofinder group showing that phospho-tau217 had the same diagnostic accuracy as CSF and PET biomarkers and able to classify Alzheimer's disease patients in a clinical setting. We also started to look at preclinical changes, so biomarker changes before symptoms. And here we see that phospho-tau231 is a biomarker that reacts very early to amyloid deposition, where phospho-tau217 is a more clinical biomarker. In 21-22, we started to see mass spectrometry methods come along, which means we could profile multiple epitopes at the same time. This means we could do disease staging and see where 231 increases first by 217, and 205 as a biomarker which reflected tau pathology. We also started to understand some of the limitations of blood biomarkers like comorbidities, that chronic kidney disease affected these levels. And on the right-hand side, we started to look in cohorts of longitudinal change. And this is very important when it comes to drug trials. And this study here, which we published in Nature Medicine on the right-hand side, showed that phospho-tau217 was really the only biomarker that was changing dynamically as you were developing amyloid pathology. And this is a very crucial distinction between 217 and other phosphoepitopes that you may hear of. Now, 4th generation biomarkers started to see a lot of competition. So on the left-hand side, you can't quite see it maybe, but we'll go into more detail. This is a round-robin study that we've hosted in Gothenburg, which showed that there were 33 different phosphoassays from vendors around the world ready to be released onto the market. So this creates massive competition, of course, and that's a good thing for science. We also this kind of illustrated the dominance of 217, and this was our principal biomarker. We also started to see in the center here this idea of phospho-tau217 occupancy, so a percentage of 217. This is a mass spectrometry method, and what this is meant to do is kind of counteract the effect of comorbidities or chronic kidney disease by doing a ratio of non-phosphorylated tau. And we started to have guidelines of use of how we would use these blood tests in a clinical setting. And maybe one cutoff to say positive or negative wasn't going to work for a biomarker, but having high risk, low risk, and unsure would be a much better way of using these. And these are guidelines that are published now that we can follow as clinicians. So coming up to the present day then, where are we now? So we have commercial phospho-tau217 assays, which is that 2024 paper that I shows at the start. And we have reported cutoffs, so labs can start using these biomarkers using independent cohorts, using cutoffs that are validated against imaging. We have combinational markers, and we have fully automated assay instruments in our laboratory, which Alamar is one of. So I've done a lot of praising here for the blood biomarker field. So maybe I could end the presentation here and say it's done. But I actually think that this is actually just the start of a blood biomarker revolution in dementia. phosphorylated tau isn't the complete answer. It's actually a very important question that we've answered, but there's many, many more now with the future of Alzheimer's disease when you think about drugs being approved, longitudinal change, the diversity that we will see in our patients, and also the multifactorial component to Alzheimer's disease. So we're looking for a personalized medicine approach. Amyloid and tau are not the only proteinopathies that develop in these brains of Alzheimer's disease patients, just illustrating here from the pathologies that we can detect through CSF. Amyloid drugs are only a fraction of what is in our drug pipeline in phase 1, phase 2, and phase 3 trials. There are a lot of different targets being developed, including inflammation and others, and we need to have biomarkers, preferably in blood, that will be able to track these changes. Can we improve the outcome in Alzheimer's disease trials? So this is the decrease of amyloid pathology after being on donanemab. This is at the group level. But what it actually looks like in reality is that some individuals don't change, some people change very quickly, some people change moderately. And we need to be able to have prognostic biomarkers to be able to identify individuals that will change rapidly or would need a different approach, or those that will not respond to these drugs. This is something we need to know before we start applying this, and this is another opportunity for biomarkers. Very, very importantly is that we need biomarkers to predict adverse effects in these trials. So there is a percentage of individuals that get ARIA, this amyloid-related measure through MRI. A lot of it is asymptomatic, but some of it is symptomatic, and this could be an issue for these types of drugs. But a biomarker to be able to detect this and being able to treat it early, or at least predict those that are more likely to get ARIA, is also extremely important. Inflammation, a very key component of Alzheimer's disease. We have emerging imaging biomarkers, but essentially in CSF and blood, we don't have anything that can track specific inflammation in Alzheimer's disease. And the most importantly is that, as I've described before, is that amyloid and Alzheimer's disease type tau is only a very small fraction of the proteinopathies that we see in dementia. There are different types of tau, which we see in CBD and Pick's disease and PSP, which are not the same type as tau as Alzheimer's disease, and we have no biomarkers for these. We have limited biomarkers for TDP-43 and alpha-synuclein and other diseases like ALS. And this is a massive challenge for us now in blood biomarkers, but also CSF biomarkers. But do we need better sensitivity? So I've talked about the need, now about sensitivity, because we're picking up a lot of good biomarkers with the methods that we have. And I would point towards phospho-tau205 as an example. We know it's an important biomarker. We know it's a late-changing biomarker that tracks tau pathology. We only really have one assay in the world at the moment, which is the mass spectrometry method, which has limitations that can detect 205. So we do need new technology to be able to detect novel phospho-tau epitopes, which could be very important for staging. And here I'm going to present our blood spot work that we've been doing. And this is, again, another case for sensitivity. We're able to detect phospho-tau217 through a finger prick test. It correlates very well with the plasma measurements. It can separate amyloid pathology negatively very well. But at the lower end, at the lower levels of these quantifications, this technology has a limitation. So using a sensitive method that Alex has described with this easy way of collecting blood would be a real game changer in blood biomarkers for dementia. So I think that we've nearly now come full circle from where I started at the start of the presentation, where we're doing discovery proteomics. We've had a decade of trying to get good assays for phosphorylated tau and NFL and GFAP, and now that's been achieved. I think we're coming back around to discovery proteomics, but with better technology and also core biomarkers to pin our results to, like phosphorylated tau217. And I think this is where, in particular in my research, this is where the NULISA platform is going to play a key role. So this is a multiplex assay. There are singleplex assays. I'm going to be mainly talking about multiplex. But one very, very important thing for us as researchers is that we want to be able to know that phospho-tau217 and NFL and GFAP embedded into these assays are of the best quality. So that was our first starting point. And this is the round robin study that we've been doing with multiple assays around the world. So on the left-hand side here is plasma, in the middle is CSF, and on the right is an area under the curve metric for predicting Alzheimer's disease. And we can see that the Alamar singleplex and multiplex assays are right at the top with these best in class. And it's the only assay that is measuring multiple biomarkers at the same time. So you have to remember that as well. And on the right-hand side, the far right, the diagnostic accuracy of this assay is equivalent to all the other 217s in the market. This is what it looks like at the individual level. So large fold changes if you use the multiplex or the singleplex compared to controls. And this is not actually healthy controls. These are clinical patients that have symptoms, but they just don't have Alzheimer's disease. Criticism of the study is that the number of patients is very, very small. So we've looked in the triad cohort with Pedro Rosa Neto, which is over 1,000 individuals with blood. And we can see that NULISA performs extremely well here, again, against the best-in-class assays on Simoa, where you have an accuracy of over 90% to predict amyloid pathology, and again, over 95% to predict tau pathology. So it really stands up and outperforms, in fact, some of the best-in-class assays, even in the multiplex format. And this is a visual way to look at those results. So this is basically correlations between the amyloid load in the brain and the phospho-tau217 in the blood. And on the right-hand side is NULISA, and the left-hand side and the middle are two competing assays. And we can see that the NULISA assay is correlating with amyloid pathology much strongly than these other assays, which is a really positive thing for us in diagnostics and also in drug trials. So these are correlations with the best-in-class Simoa assays and the tau we've talked about, but in the middle here we have neuroinflammation in GFAP and NFL, and as Alex has always described, the R-squareds of these are extremely good, and they're giving us similar results, again, but in a multiplex format. So now we've shown that the biomarkers that we expect to work very well in blood do work extremely well on the Alamar platform. What about everything else that it measures at the same time? So I've just got 3 or 4 novel case studies that I'll finish with. The data that we are generating in Gothenburg, we have an Argo instrument installed there, and we've been generating some results. So here are a few case studies and challenges that I think that the Alamar and the NULISA system are going to help us with. So this is Elmo's work. He's a PhD student here. We used the CNS disease panel here in plasma. And the first thing we're looking at is mild cognitive impairment. So this is a prodromal dementia stage. 50% of these individuals are usually Alzheimer's disease, but it's very difficult to understand the underlying pathology in these individuals here. So what we're looking at is, does these individuals have amyloid pathology or not? And this is a volcano plot to see which biomarkers are important, either upregulated or downregulated. So this is completely unbiased proteomics in this 120 disease panel. We see that phospho-tau217 is clearly the most important biomarker in the top right-hand side. A-beta-4240 is a downregulated protein as you expect, and we can see 231 and GFAP as well. But a lot of downregulated inflammation proteins and microglia proteins are also emerging here, which could be extremely important for disease staging and personalized classification. If you look at early tau deposition, which is also an important metric, so in this volcano plot, everyone has amyloid, but we're wanting to see who's starting to develop tau, and tau is very closely linked to the onset of symptoms. And again, the volcano plots show phospho-tau is the best biomarkers in GFAP and neurofilament light, like we'd expect, but a different profile of microglia proteins and inflammation proteins are downregulated, and some other synaptic proteins are upregulated in these areas. So again, these are biomarkers that would add to our classification models on top of phosphorylated tau. Sticking with plasma, but now looking at Lewy body disease. So we have a good biomarker in CSF using RT-QuIC or a seeding assay, which is a replication of alpha-synuclein. Now we're looking at the plasma of these individuals where these replication assays don't work so well. And we can see here that Lewy body disease versus Alzheimer's disease, we can see that phospho-tau is on the left-hand side, so that's with the upregulation with amyloid and Alzheimer's disease, but some important biomarkers here on the right-hand side, which are maybe indicators or sub-indicators of Lewy body disease. On the right-hand side is a box plot showing the aggregated alpha-synuclein assay in plasma. It's a small decrease. It's a significant one. This is actually log fold change, so it's not. So it's a lot larger than how it appears on the graph. But this is the first time we've actually seen a working aggregated or seeding assay working in plasma. So this is something we've started to look at in larger individuals or larger groups of individuals. Sticking with plasma, now looking into more genetic types. So progranulin is a most common genetic form of frontal temporal dementia, and is often used as a model to look for biomarkers for sporadic types of frontal temporal dementia. And here the results are really remarkable. This is just 20 individuals with the mutation and 20 individuals without. And you can see the number of significantly upregulated proteins in these individuals. Neurofilament light is at the top, and this is exactly what we'd expect. But we're starting to see synuclein and synaptic biomarkers. Interestingly, the TDP43 and SOD1 are also proteins that we would expect to be associated to an FTD type dementia, but we've never been able to profile them in plasma before. So the last two case studies I'm going to look at in CSF, and I've been talking about blood the whole presentation, but when you don't have a target or when your target is not so clear, I still think that the relevance of biomarker discovery in CSF is extremely important. And I want to illustrate this with phospho-tau217. So this is a paper that we have published. The title of it is of Equivalence of phospho-tau217 in cerebral spinal fluid and plasma. And because it's my paper, of course, I believe it's true. The diagnostic accuracy between plasma and CSF is almost identical. And this has been shown by many individuals. But I want to show you a different way of looking at the difference between plasma and CSF. And this is extremely important for discovery. Again, this is this round robin study that we have done with all the phospho-tau assays. And this is the plasma results. And this is fold change. So how many times increased in blood is this biomarker increase compared to controls? And again, the accuracies are very, very high. They're equivalent to CSF. But what about the signal of that protein in blood? If we superimpose the CSF on top, we can see that the CSF results have such a larger signal in this biofluid than plasma. It's a much easier metric to measure. So if you're looking for new biomarkers, your signal is going to be higher in CSF. And that's an obvious statement to make, but sometimes we jump to blood and we may miss something because the signal is not there. So I think it is very important for us to still do discovery proteomics in cerebral spinal fluid. And here's one example. This is actually the inflammation panel we've used, the 250 panel. And this is Ilaria's work. And what she's looking at is inflammation proteins that are predicting inflammation imaging in the brain, which is this PBR, which is a TSPO binding tracer. And we're starting to see some really nice inflammation proteins that can predict inflammation in the brain. And this is so vital for Alzheimer's disease. I think that inflammation is a key missing component which connects amyloid and tau and important for our clinical trials. This is CSF now, but looking at amyloid pathology. And again, we can see phospho-tau is a key biomarkers, but a lot of other significant biomarkers coming up below it. The right-hand side is basically an enlarged version of the left-hand side, and we can see, again, key biomarkers we haven't considered before, but also ones that we do know about, like synuclein beta, 1433 and FAB-B3, are biomarkers that we now might want to consider in our diagnostic models and maybe pursue in blood as well. And looking at tau pathology in a similar way, again, phospho-tau is our key marker, but a whole host of different biomarkers that are changing when tau pathology is developed. And we can see this in a multiplex form, a lot of synaptic markers, and again, synuclein, 1433, and neurofilament light, and others. And lastly, I want to finish on this. I think this is a simple idea, but I think it's a really effective one. And this is measuring ApoE protein in the blood. Embedded into the multiplex is an ApoE assay. It measures ApoE as a whole, or ApoE4. And basically what you can do with these protein levels is do a equivalence of a genotyping. So is there an ApoE4 allele present or not present? And this is the most common sporadic genetic risk factor for Alzheimer's disease. And this is a prototype assay, and this is on a singleplex ApoE measurement. So this is not available. This is a prototype that we've been lucky enough to collaborate with Alamar on. And at this level, you can do full genotyping. So you can actually divide between a heterogeneous or a homogeneous E4 carrier. So a really exciting way to genotype individuals. And of course, that's already embedded in the multiplex results that you get. So to conclude my talk, we've spent a decade doing plasma proteomics and come up really with a biomarkers that we knew would work very good from CSF. And we've landed on phospho-tau217, and this biomarker is already approaching clinical use worldwide. But it's not the answer for everything in Alzheimer's disease, and definitely not the answer for other neurodegenerative disorders. We need biomarkers that are going to tell us about treatments, about individuals that are on those treatments, and about the co-pathologies that develop alongside Alzheimer's disease. So we're starting to use the NULISA-seq because it includes 217, NFL, GFAP, and ApoE genotyping synaptic biomarkers, but it has a whole host of combination of proteostasis, energy homeostasis, skeletal abnormalities, DNA and RNA deficits, inflammation, and many more, which may give us a whole host of important information that we can put around phospho-tau217 and NFL to come up with a better prognostic and deal with the challenges that we have when we are starting to have effective drugs. So as I just said, I think this is a very, very important method for new drug targets in Alzheimer's disease and other developments, novel biomarkers for our future challenges related to AD progression, other disorders. And of course, I mean, I've been talking about dementia, but any other disease that you are essentially studying, this is a great way to start a hybrid between targeted discovery proteomics. So I want to thank the Alamar team. We've had a great collaboration with them to generate some of this early access data. This is the team in Gothenburg and our collaborators on the right-hand side. And Alamar haven't told me to say this, but we really like the technology. We're very keen to try it in multiple disorders. And if you would like to collaborate, we have the Argo system in Gothenburg in Sweden. We'd love to do some studies with anyone that's interested. And thank you very much.
Thank you, Nicholas and Alex, for your informative presentation. We'll now start the Q&A portion of the webinar. If you have any questions for our speakers, go ahead and submit them now. Go ahead and get started. We have a few questions from the audience already. So I'll start with this one. Is it possible to run libraries on other NGS platforms or only Illumina? Thank you for the talk on a great tool.
Right now, it's only available on the Illumina sequences, the NextSeq or NovaSeq series of sequences, but we are working to enable other sequencing technologies as well.
Great. Thank you for that. We have another question here. Are the correlations between neurodegenerative diseases and gray matter atrophy?
Yeah, that's an interesting question. So we actually see that different biomarkers correlate differently with different types of atrophy. So a protein like neurofilament light or GFAP, they're actually more likely to correlate with something like white matter atrophy. So more of the white matter axons in the brain, whereas phosphorylated tau is more likely to correlate with grey matter atrophy because that's where the pathology kind of develops. So depending on what biomarker you're looking at, there is different associations, but there is definitely a strong association there.
Great, thank you so much for that. We are coming up to time, so we have time for about one more question. So I'm going to say our final question for today is there an effort to recruit and include people with trisomy 21 in clinical trials?
Yeah, I can take that one. So Down syndrome is a
now considered really a familial genetic type of Alzheimer's disease. Because these individuals live longer because of better care, we do find that there is almost a full penetrance of them developing Alzheimer's disease. So the theory is, of course, that they will benefit from these anti-amyloid trials as well. So they're not being recruited within Alzheimer's disease trials. They are being recruited as their own population, but that is definitely ongoing. Something like the Alamar platform is something really interesting for Down syndrome, because we know that the Alzheimer's disease biomarkers work, like I talked about. But of course, there is a different element to Down syndrome, like inflammation, which could set it apart from a different aspect to Alzheimer's disease. So this inflammation plus AD biomarkers in Down syndrome is going to be extremely important to study.
Wonderful. Thank you both so much for taking time to answer these questions from our audience. Well, Nicholas and Alex, again, thank you so much for your time today and for your important research. A big thank you to Alamar Biosciences for sponsoring today's webinar, and to our audience for joining us today. Questions we didn't have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until March 6th, 2025, and LabRoots will send you an email letting you know when it is available to be viewed on demand. We encourage you to share that email with your colleagues once you receive it, who may have missed today's live event. Until next time, goodbye.